Yeah, I think I have all the time until lunch, uh, which is probably half an hour. But I started feeling hungry right away. So I better finish it also fast for my own welfare. Um, I'm running a bad throat. It's not related to coronavirus. Others, none will be left in the audience. And what I would like to do this morning, actually it's a conspiracy to put me at the end of the series of lectures. And that conspiracy is not because of my age, but perhaps to do with the Microbiome Connect. And in the taxa of whatever we have listened to this morning, I think this is in the very, very low, probably underground. And what I hope to do in the next few minutes is to sort of reawaken in your minds the importance perhaps of microbiome, and maybe have a little bit of converts in you when you go back to your lab and ask yourself, oh, this could also be an added dimension to the issues that we are now looking at in various contexts. So I hope this works. Daya had a horrible time with it. So Professor Bawa and I met in Sydney, the University of Sydney, I think nearly 40 years ago in 1981, when we both had been there to attend the International Botanical Congress. And I should say that I was quite impressed by the, I think, 3 or 4 consecutive evenings, simply because he took me out for dinner and I was broke at that time. And since then, since 1981, we have been, of course, in touch. It's evident. And what has happened, this, I hope this works, right? Otherwise, it's quite annoying. You want to do something about it? Unless it's a touch screen that I move it. Just one click. I'm only doing one at a time. Yeah. Okay. So what has happened since then was that I probably have not become wiser of my age. I am 40 years older, and I still am battling the problem that you see on that far side of the screen. Let's spell the damn fruit apple, which is the curse of all human population. But as speakers after speakers have told this morning, Bawa in this last 40 years has actually gone miles ahead, and you know all the laurels that follow him. And the most recent, of course, is, what's the most recent? I don't know. I sort of lose touch of that. But nevertheless, during these 40 years, one thing that has been abiding is that our relationship has been very, very warm. I can't say hot. And for those of you who are data scientists and want to always vouch on data, I have data to show that. And what is that? That's the data. Look at the data. Exactly for the period 1980 to about now, the anomaly of Earth's surface temperature, as well as ours probably, has been monotonically increasing. Now this is not a sort of made-up graph. It is just a truncation of a graph that climatologists know better, like Jagdish, somewhere sitting here. I've just simply sort of snipped off that graph for the previous one. The point I'm trying to make is that during this period, I know it's coincidental, don't read any causation and all this stuff. We have no such overarching sort of power to change global surface temperature through our relationship. But all the same, there's a monotonic increase of temperature. Now, this has been, by the way, I'm not a climatologist at all. My only love with climatology is been just last 2 days preparing for this talk. And so if you have any questions on climatology, you should ask Jagdish and Sharath. I think they are both available here. They are the best climatologists, at least that I know of. But nevertheless, like many of you who have had a fleeting glance about what climate can do and not do and all this stuff, we are all witness to this iconic pictures of what climate change can do, such as the melting of ice sheets over the Arctic, and of course the penguins not finding any place to stand. So also the polar bears struggling with no ice. And I have been always impressed, if impressed is the correct word to use, for this beautiful animation of what has happened in the Glacier Park of Montana. Starting 1910, there were about 150 odd glaciers in this place, but today I am told it is about 15, not 30. This slide is dated, and I have not been to Montana. Those of you who have gone there on a frequent sort of at intervals of time may have been noticing this one, but a very beautiful simulation of the, of that shows that the glacial cover of this Montana National Park has been receding at a very, very, very rapid rate, so much so that it is predicted by, as you will see, by 19 or 2000, we have lost all of the glaciers from this park. And of course, the other vegetation is going to sort of take hold of those areas. Now, we have in our own country very recently some indication of what is going to happen in the third pole, that is Mount Everest. And this some of you would have again seen, again spans coincidentally the period of our relationship from 1980s to 2018 over a certain range of Himalayas, where you find that there has been an irreversible glacial melt, and all of this is gone forever, even if we stopped the sort of rise of temperature globally and for part of the reason that we are talking about. And there is very much, I would say, great evidence to indicate that glacial lake progression is on the rise each year, and this is between 1989 and 2016. But as I told you, I'm not a climatologist guy, but as a person who is interested in plant stress physiology, one of my interests has been in trying to look at plant adaptations to climate change and how do they adapt to climate change, not at the scenario that we have been talking of in the terms of 30, 40 or 100 years and so on and so forth, but in more proximate sort of scenario such as drought, such as salinity and so on and so forth. And when you look at literatures on plant adaptations to climate change, and if you now look and strategize of how plants would now strategize themselves and adapt and be resilient to some of these changes, there are broadly 2 or 3 approaches that I think plants take. One is that the first thing is to take flight, just like Agra and Arjuna brought the Indians from Wuhan. That's the recent news, right? And so you take flight, doesn't matter how. It doesn't really matter. And if you look at literature that dots some of this area, you would find unmistakably examples of some plants, I won't go into the details of all of that, expanding their ranges and moving northward just to escape the warming of the lower latitudes, and so going off into the northern latitudes. And this is some sort of a common phenomena across the world and is well recognized. Though of late, in recent years, there has been some sort of question about how much of this attribution is actually due to climate change. So that's something that we can't discuss right now, is a little bit of teasing out the confounding variables. But then, this is one option. But as a part-time geneticist and evolutionary biologist, I believe just taking flight alone is not going to be really a solution, because if these guys were to move up, they have to counter a number of other, they will save themselves from temperature, but they will have to counter themselves against a whole other oddities that they have never been adapted to. So aren't these populations going to be maladaptive when they move and actually be the worse off than staying here? What is the solution? The solution is have sex and then take flight. Now, why do I say that? By that, we are increasing the genetic variance in the progeny and therefore enhancing the probability of survival of those that would move northward. Now, this is a well-recognized feature, at least in evolutionary biology circle and population geneticist circle. And one of the good examples that Professor Ganesh and I have been working is about this species of plant called water hyacinth. And all of you have seen water hyacinth. Actually, you would have seen these water hyacinths not flowering. And these don't flower as long as they are sort of floating. When they are floating, they multiply by simply doubling by the stolon production. But the moment they get fixed in the soil because of water depletion and overcrowding, they have sex. And that means they start flowering. They seed and the seeds are put out. That is, they move out. Thanks to the genetic recombination that happens, the progeny are better off into an alien environment, and so you have an adaptability. So one mantra is, when in fix, have sex. Now, some of you, please don't take it literally. These are all, you know. The third option is to fight it out, adapt. Now, here is what I have been sort of introspecting of late, of how do plants adapt to climate change, given the fact that climate change is a very, very fast process in nature. That is the anthropogenically created climate change. Now, look at this. In just a matter of 100 years, we are climbing Mount Everest in terms of global warming. Now, evolutionary biologists will tell you that evolution does not work at that pace unless and until it is also implied with a certain selection pressure, selection intensity. And so, just like the zebra can't develop claws overnight, we don't expect plants to defend or allow defenses, especially at the cellular level, tolerance to be built up to something that they have not been used to in their evolutionary time. I might be wrong in this conjuncture, but theory states that to now get adapted, you need to sort of have that evolutionary time to now reassemble your genes and then therefore become adapted to the new landscape that demands the adaptation of it. Now, this is where I come to the last and the important part of my talk of the microbial connect. Now, thanks to the NIH-sponsored Human Microbiome Project in the last 10 years, we now have an explosion of knowledge on how microbiome can influence a huge bunch of phenomena in plants, in animals, and all organisms. And of course, microbiome themselves. For example, the microbiome today is known to play a whole bunch of role in fact, deciding whether one is obese or not. Not what you eat, not your genetics, but what the gut microflora is there in one's gut. Children born through C-section are or have a very different microbiome compared to those that are born otherwise. And because of that, C-section babies are supposed to be immune compromised. And so we have a whole medical therapy in trying to now make them competent by having them get the mother's microbiome through various sort of medical treatments. And today, what is more spectacularly known is that our genome sitting in our cell is vastly outnumbered by the microbial genome, and because of that, you have a question of who's actually ruling you, who's actually modulating our own responses. And very popular book by Alan Collins says, are we just 10% human? Considering the fact that if we have 100 trillion cells, only 10% of that 100 trillion cells belong to us, rest of it 90%. If you now really get this into your minds, as it has gone into my mind, I often do this. I do not know who's giving the talk. And should I also credit the talk by saying the senior author of the talk is Microbiota of Shaanker. The junior author is that 10% guy, I don't know where it is. And of course, the microbiome of Shaanker is in the gut and brain of Shaanker. Now, having said that, there has been a paradigm shift in how we look at organisms and their adaptations. And the paradigm shift is, until 10, 15 years ago, the geneticists, plant geneticists and animal geneticists would vouch that all of the phenotype that we have is attributable to the genotype of either the plant or the animal, but no more so. Today, we are intimately associated with the microbiome, so much so that you cannot shed off that from ourselves. And so, we talk about the microbial genome as some sort of a component embedded in our genome, and it's better to call our genome not as host genome, but with the microbial genome as the hologenome, which then interacts with the environmental metagenome to produce what we call as the phenotype of any plant, which renders ultimately the adaptability to the plant. So, in genetics terminology, the phenotype is just not the interaction between the genotype and the environment, but it is the interaction between the genotype of the host, the microbial genotypes, and the environment. So, you have an additional component over there. So, microbiome is the new fashion, and I am fashionable. Do I look fashionable? So, I am converted. Now, that brings me to the last part of the talk about how do we now take this big leap of asking, can or does the microbiome have in it the capacity to modulate in a manner that can actually make plants more adaptable to climate change. Now, in this cell, this part is a plant cell, and that is a microbiome, microbe cell or microbiome, if you have to now put all of that. And normally in us and in the plant, the microbiome, it could be a bacterial cell, it could be a fungal cell. They reside in what are called as apoplastic spaces, that is in the intercellular spaces. So, this is the intercellular space, this is a cellular space. They don't enter the cellular space. But in the intercellular space, the microbiome is also exposed to the same adversity as the host cell is, for example, to climate change, to salinity, to high temperature, to drought, to oxidative stress, what have you. But simply because a bacterial cell is going to multiply many times faster, and of course, going to go through the spontaneous mutation, one would expect that the bacterial cell would adapt faster to climate change events compared to what a plant cell would undergo. That is number one of the hypothesis. Then the number two of the hypothesis is that if it adapts faster than the plant cell, because of the signaling between the apoplast and the symplast, there are mechanisms that we could now look at in which the bacterium can now transmit the adaptation that it itself has acquired to climate change into the host cell and make the host cell also adaptable to the adversity much faster than the host cell would otherwise evolve with. Now, this is a mantra where you are now accelerating a process by which plants become adaptable to climate change events not by their own genome, but by the extended genome, as Richard Dawkins mentioned, not the extended phenotype, but I'm talking about an extended genome, and that extended genome comes from the bacterial cell or the fungal cell that are floating in the apoplastic spaces. Now, is there any evidence for that? I'll give you two examples from our own lab in the recent past that we have tried to now look at. There are other examples published in literature, but I shall relate two of those from our own lab. Now, what is this example? This example is a microbiome analysis of the plants of Thar Desert. And of course, you expect that plants of Thar Desert to be adapted to thermotolerance. And so we went to the Thar Desert and sampled many of the plants that you see in the Thar Desert, so that many of them. And from there, isolated the microbiome, in this case fungal microbiome, from them. They are called endobiome, will not bother with the jargon of that, and found that many of them were actually tolerant to high temperatures. Normally we sort of grow the fungus at about 25, 28 degrees, but then at elevated temperature of 45, you find that many of the fungus are able to survive without any great depreciation of the growth. If you like to look at this last fungus, it's called LAS6. It's a fungus called Chaetomium thermophilum. It's named after its ability to tolerate high temperature stress. And the same fungus is also available in Yellowstone National Park area. Now, if you look at this fungus, there is no appreciable decrease in the growth of this fungus between 28 and 45, does absolutely perfect. The question is, if this microbiome that is sitting in my apoplast, if I can push this into a plant which has not witnessed or experienced 45 degrees centigrade, can I make the plant tolerant to 45 degrees centigrade? So you now take this fungus, by some technique, you can now push this fungus into the apoplast of, let's say, in this case, rice seedlings. And what you find is, when the rice seedlings are exposed to 45 degrees, without the fungus, they perform poorly, but when with the fungus, they grow normally compared to the ones that are growing at 28. And all of this in just about 3 or 4 weeks' time. You have made a sort of magic in the laboratory just by transferring the fungal cell into the plant cell and making the plant cell, and there's absolutely no change in the nuclear genome or the chloroplastic genome or the mitochondrial genome of the plant, but only what has been done is you have put this fungus into the apoplast space of these plants and made them that at 45 degrees. Another example is the microbiome from a mangrove plant. Now, this is called a salt brush plant. If you now take this plant, clean it up, and then just taste the leaf of this plant, you will find that it's very, very salty simply because it extrudes salt out of its salt glands. And from this plant, we again pulled out the microbiome, and one such example is this one. And what you can do is, you can repeat the experiment, especially this very important in the IGP area, the Indo-Gangetic Plain, where lots of soil is sodified, as you see here. And you can do the same experiment of pushing it into rice seedlings or rice plant. And what you see here is, these two plants are the ones that have been amended with these fungus, whereas this is a control plant. What you do, or what you see is, in one generation, a salt-sensitive rice plant, this is sensitive, has been made into salt-tolerant rice plant, all in just within the space of one generation. And this is transgenerational, so if you collect the seeds out of this plant and this plant, in the next generation, they are going to be salt tolerant. So you actually now started a breeding improvement program, not by conventional breeding of the salt tolerant and sensitive variety of rice, but only by sort of putting in the microbiome. So what I'd like to conclude here, and that is in the sense we are at very early days in our lab, is that where is the big leap when we now talk about community responses to climate change in plants? the question, a big question is, do microbiome enable plant adaptations to climate change in nature? Or do microbiome or can microbiome accelerate the plant adaptation to climate change in nature? Now, my gut feeling is it must be by simple extrapolation, and what is that we are trying to do today is now working at certain parts of the country where we are looking at the microbiome of plants at different altitudes as a proxy of the climate sort of variable, climate change variable, and looking at a whole metagenomic library of plants which are available all through an incline, and asking if these plants harbor a metagenome library different from something at the mid altitudes and high altitudes, and do some sort of transplantation of the community of microbiome and ask if by doing so we make plants adapt to temperatures, in this case temperatures, faster than otherwise. So with this, I'll conclude my talk. I'd like to thank Suri, who is a mycologist. Incidentally, he is Dr. Sukumar's teacher. Ravikant here, Dr. Nataraj Karba, my collaborator at GKVK, Daya, who is collaborating with some projects on this one, and Ganesha, who contributed to when you are in Fix Have Sex. So if you have any questions, catch him. I don't have answers for that. And my lab. And this work was primarily funded by the Department of Biotechnology under the NER Bangalore Region Chemical Ecology Grant and a couple of ICR grants. Thank you very much for listening.